All right, we're finally starting our genetics unit, one of the most fun units of the entire year of biology. And we're going to start out with the very basics, Mendelian genetics. What are the basic ways that we inherit information from our parents? So that's all that genetics is, understanding how traits get passed on from parents to offspring. We can use these rules that we're going to see later in the presentation to actually predict what characteristics an offspring may have. Traits or characteristics are just the physical things we notice that get passed on. So, do you have your mother's eyes? Do you have your dad's curly hair? And it all started in the 1800s with an Austrian monk named Gregor Mendel. He really wanted to be a teacher, but back then that required a lot of schooling, which was incredibly expensive. And his parents were farmers, so they didn't quite have the money to send Gregor to university and to learn how to be a teacher. So he instead became a priest, which was a very common thing back then because it allowed you to still get some education and it offered you a more comfortable life compared to the hard life of a farmer. So because he came from a background of a farming family, Gregor Mendel was the one that tended the gardens that fed the entire monastery where he lived and worked and taught. So it was while he was tending for his garden that he started to notice some patterns in how traits were passed on. He noticed that offspring were always inheriting traits from their parent plant, and he wanted to study how are these traits inherited? Why did one trait get inherited over a different trait? So in particular, he picked pea plants. It might have been because pea plants are really good for the soil. They add nitrogen back to the soil through nitrogen fixation, but it may have just been because he liked peas. Anyway, it ended up being a really good decision because, as we'll see now, peas have very specific characteristics and they have two versions of these traits. He looked at a bunch of traits for the seed, and seeds can either be round or wrinkled. They can either be yellow or green. The flowers can be white or purple. The pods have a couple different characteristics. They can have a full appearance where they look kind of inflated, or they can look constricted, like you sucked all the air out of them. You can look at the color. It can be a yellow pod or a green pod. And there's even characteristics about the plant overall. It can be a tall plant or it can be a short plant. The flowers can be placed in the middle of a branch, or they can be placed on the edge of a branch. So the fact that there was two very specific options for every trait actually made pea plants very good to study. So Mendel very strictly controlled how he pollinated these flowers. He cut off parts of the flowers that produced pollen. Then he would take a brush and he would collect pollen on the brush and put the pollen on the flower of the plant that he wanted to breed with. So he would very specifically take the pollen from one plant and put it into the flower of a different plant. And those were the two parents, the two parent plants. Now, he decided to take true breeding plants. These are plants that have identical traits to their parents. So in our example, this white flower had two parents that had white flowers. The other flower was purple. So this one had two parents with purple flowers. And when he took these true breeding plants, the ones with the parents that had the same traits as them, he found something fascinating. All of the offspring had one particular trait. They would always have purple flowers. He called this the F1 generation. F refers to the Latin word filial, which means sons or children. So it makes sense that this is the first generation of children. So the F1 generation always showed one particular trait. He did this with all of the other traits and found the same exact pattern of inheritance. He would cross a tall true breeding plant with a short true breeding plant, and in the F1 generation, he got all tall plants. So he called this stronger trait the dominant trait, and we still use that word today. Whatever trait overpowers the other is the dominant. So you can see in the F1 generation, this particular row shows all of the dominant characteristics. So we've got tall plants are dominant to short plants. Green pods are dominant to yellow pods. The inflated pod is dominant to constricted. Round and yellow peas dominated over green wrinkled peas, and so on and so forth. So Mendel decided that that wasn't enough. He wanted to cross-pollinate the F1 generation. He wanted to take all of the offspring from that F1, those tall plants or those purple plants, and make them cross-pollinate each other. And then he created, as a result, the F2 generation, the second generation of children. And as a result, he had some very fascinating results. 3 plants were tall, and 1 plant was short. And the same thing happened every single time that he performed this F2 generation mix or cross. 3/4 or 75% of the plants had the dominant trait. 1/4 of the plants, or 25% of them, had the recessive trait. So now that we notice that there are these patterns to inheritance, we can predict what might happen if we cross 2 plants. But first we have to know a few key terms. First is this term phenotype. This is what we use in genetics to say the physical appearance of the trait, the purple flower, the tall plant, the green seed, things like that. The way I remember it is the pH in phenotype matches the pH for physical. The next term that we use in genetics is this term genotype. And this is the genetic makeup of the trait. What are your actual genes saying? The way I remember this is the GEN for genetic. And we're going to talk a little bit more about what these letters of the genotype mean in a moment. So the genotype is the genetic code for the proteins that we see in the phenotype. We talked about this a little bit when we went over transcription and translation. We said that DNA coded for RNA, and that coded for our proteins. That was the central dogma of biology. Well, now we can see that the proteins are just a stand-in for our traits. So the DNA, our genetics, codes for our traits. Genotypes code for phenotypes. Now, genotypes are commonly represented by 2 letters. The dominant trait is represented by a capital letter, such as like a capital A. The recessive trait is represented by the lowercase letter. And we always have 2 letters to represent our particular genotype. Why do we have 2 letters? Why don't we have just 1? Think about it and answer the question, and then come back to the video. So why do we have 2 letters? Well, we have 2 parents. We get 1 letter, 1 gene from each parent. If you look at the picture to the left here, we have our fly, and he has 2 copies of these chromosomes. He gets 1 copy from 1 parent who maybe has a dominant phenotype, and maybe we get the gene from the other parent that has a recessive phenotype. So this particular fly's genotype would be big A, little a. It's customary to always put the capital letter first. So alleles are just the copies of our genes. It's one of the 2 copies that we have. You have the dominant allele, or you can have the recessive allele. So in either case, it can be dominant or recessive. You can end up with 2 dominant copies, or you can end up with 2 recessive copies. It is completely based off of essentially random mixing of your parents' genetic information. So the dominant allele is always going to be the capital letter. Now, the trait that is associated with that dominant allele will show up even if there's only one capital letter. So it's going to show up if I have big B or big B little b as my genotype. Because in this case, the capital letter is always going to overpower the lowercase letter. So this particular organism that's big R, little r, for whether the P is round or wrinkled, is going to have the round phenotype. This wrinkled phenotype is going to be hidden by the dominant round allele. If you ever want to see that recessive allele trait, you have to have 2 copies of that recessive allele. You have to have 2 little letters as your genotype. This is Mendel's law of dominance. Now, our genotypes have their own specific terms based on how we combine them. The term homo in science means the same, and the term hetero in science means different. So you can be homozygous, where you have 2 of the same letters, such as 2 capital A's or 2 little a's, or you can be heterozygous, where you have a mix of capital and lowercase. If I'm homozygous dominant, I'm homozygous, so I'm going to have 2 of the same letters, and it's dominant, so it's going to be the capital letters. So this would be a case of big A, big A. Homozygous recessive means I'm going to have little a, and hetero means different, so this would be my big A, little a scenario. I think I have another example that I wrote on here. Yes, so homozygous dominant would be 2 capital R's. Homozygous recessive would be not big R, little r. It would be little r, homozygous recessive. And then heterozygous would be big R, little r. So let's look at some of these examples here. If you are homozygous dominant for eye color, where the dominant big B means brown eyes and the recessive little b means blue eyes, what would be your genotype? What would be your phenotype? Write that down in your questions and then come back to the video. So your genotype would be big B, big B. You are homozygous, which means the same, and dominant, which means it's going to be the capital allele. And your phenotype would be brown eyes. Now, what if you had the heterozygous genotype? What would that look like, and what would your phenotype be? Well, your genotype, if you are heterozygous, means you would have 2 different letters. So you would have the big B and then the little b. And your phenotype would be brown eyes, because that big B allele is going to overpower the little b allele. And the last question, if you were homozygous recessive, what would your genotype be and what would your phenotype be? Well, your genotype would be 2 small letters, because you're homozygous, so it means the 2 of the same letters, and it's telling you that you're recessive, so you have 2 little b's. And that would make you have the recessive phenotype of blue eyes, because there's no big B to hide my blue eye trait. So now let's get into how we can predict these crosses. We need Punnett squares to do that. So the Punnett square is just a tool that helps us see all possible combinations of alleles for the 2 parents that we're crossing. One parent's alleles go on one side of the square and the others go on the other side. It doesn't really matter which parent is on which side. You can have the mother on this side and the father on this side, or the mother on this side and the father on the top. But you always put one of their 2 letters per row or per column. You don't want to put 2 letters inside that column. We're just using 1 letter per column. So let's do an example here. Let's say one parent has capital T, they're homozygous dominant, and the other is homozygous recessive, little t. So I'm going to say that the mother's genes are homozygous dominant and the father's are homozygous recessive. How would I do this cross? I would take the big T from my mom and move one into this particular square next to her row. I can also move this T into the other part of the row. I'm going to do the same with the bottom row. And now I've filled it all with my mother's alleles. I'm going to do the same with the father's alleles now. That little t can move in. And we're going to do that for the entire row. So I would end up with big T, little t mixes for all of the offspring. They would all have the same genotype and the same phenotype. Let's do this one again, where we're crossing two heterozygous offspring. So we're going to have one parent be big T, little t, and the other parent is also big T, little t. So when we do this cross, we can bring the big T down, the little t down, and then we can take the mothers going across. And this one we get a little bit of a mix. We see that there's going to be one part of the offspring that have big T. Two parts of the offspring can have big T, and the last one can be little t. So we're eventually going to convert these to percentages and see what's the likelihood of me getting a certain type of phenotype or genotype based on a cross. So this is actually my ratio of all of my genotypes. I have a 1 to 2 to 1 ratio of genotypes. So as a review, we would take the parent generation and cross true breeding plants, one that's tall and one that's short. And in the F1 generation, we would get all tall plants. They would be heterozygous. They would all be a mix of big T, little t, based on what the parents could give. Then if we took those big T, little t heterozygous offspring and crossed them in the F2 generation, you would see the pattern of 3 tall plants and 1 short plant. The genotype pattern would be 1 plant is homozygous dominant, 1 plant is 2 plants are heterozygous, if I can read properly, and 1 plant will be homozygous recessive. As I said, we're going to use this now to calculate probabilities. I can now say that there's a 25% probability that that cross will make a short plant. It's kind of like the idea of what's the probability if I flip 2 coins and they both land on heads. So if you look at this Punnett square here, I have a gold coin and I have a silver coin, and I'm going to flip both of them at the same time. What's the probability of me getting heads for both coins? So we have heads. I'm going to use a different color here to make it show up a little better. And tails. Oops. That's going to be represented by a small h. Heads and tails. When I complete this Punnett square, I can now get a nice idea of what's going on. The probability of me getting 2 heads is only 1 out of 4, which is 25%. So we have 2 different probabilities that we can calculate. A genotypic probability, what's the probability of a certain genotype occurring, such as capital H, or the heterozygous genotype, and what's the probability of the phenotype occurring. such as what's the probability of me getting 1 heads, 1 tails of either coin? Well, that's going to be 50% because I had 2 instances of one of them being heads and one of them being tails. I had 1 instance of 2 heads and 1 instance of 2 tails. So it would be 50% of a heads to tails mix, 25% of a head-to-head mix, and 25% of a tail-to-tail mix. We can represent these as percentages, or we can represent these as the ratios. So I'll have, for example, 3 tall plants and 1 short plant, or 1 big T, 2 big T, little t's, and 1 little t. So let's take a look at probabilities while doing a Punnett square now. So we're looking at the probability of a heterozygous tall plant crossing with a homozygous recessive short plant. So this is not telling you what your genotypes are in the sense of giving you the letters. You have to figure out the letters on your own. So a heterozygous tall parent, heterozygous means different. So this would be a big T, little t parent. A homozygous recessive parent is the same for the recessive allele. So that means 2 little t's. So it doesn't matter which parent it goes on here, the mother or the father. This is just trying to get through your head that you get 1 set from each parent. So in this case, when we complete the Punnett square, what will be the probability that they produce a heterozygous plant? Well, this one here and this one here are heterozygous, but these 2 are not. So the probability that they produce a heterozygous plant is 2 out of 4, or 50%. Now what's the probability that they produce a tall plant? So you could see that my first one was asking about the genotype. This one was asking about genotype. This second question is asking about the phenotype. What's the physical appearance? So this plant has a tall allele, so it would be a tall plant. This one has a tall allele, so this would be a tall plant. Both of these lack that tall allele, so they would be short plants. So in this case, the answer is going to be the same. I'm going to produce 2 out of 4 of the plants as tall plants, or 50% will be tall. So I want you to try to do this one on your own. Answer the question to the right, and then come back and we'll do the solution together. What is the probability that a homozygous dominant purple flower and a homozygous recessive white flower produce a homozygous dominant plant? And what's the probability that they produce a white plant? So homozygous dominant, let's use purple for this one since it's talking about purple flowers. Homozygous dominant means the same, and it's the dominant allele. So we're going to have 2 big P's here. And then we have a homozygous recessive white flower. That's going to be 2 little p's. So I have one parent that has homozygous dominant and the other parent that has homozygous recessive. When we do that cross, this is what we end up with. So what's the probability that we produce a homozygous dominant plant? That means what's the probability of us getting a plant with 2 big P alleles? Well, I don't see any of that in my Punnett square. So the probability that we produce a homozygous dominant plant is 0 out of 4, or 0%. Now what's the probability that they produce a white plant? Well, in this case, I have alleles that are dominant in every single case. So we're going to have purple flowers in every single case. So do I have any white plants at all? Once again, 0 out of 4 are going to be white, or 0%. So Mendel ended up discovering 3 laws of genetics. The law of dominance, which says one allele is dominant over the other. The law of segregation, that says that the alleles are going to separate randomly, so I'm going to pass on a random allele to my offspring. And actually, Mendel didn't really discover this last one. This is when we really started learning about DNA. But when meiosis is occurring, during meiosis 1, when I have all of my homologous chromosomes lining up on that metaphase plate, they line up randomly. I'm not going to get all of one parent's chromosomes on one side and all of the other parent's chromosomes on the other side. It's going to be a random mixing every single time they line up. So now, Mendel's peat plants were actually a very rare find. It's hard to find traits that actually follow Mendel's strict laws of inheritance because, let's face it, life is a lot more complicated than just dominant and recessive. We have the environment that affects it. We have special kinds of traits that only get passed on if they're on an X chromosome or a Y chromosome. We have things called incomplete dominance and codominance. So he picked some good traits to give us the basics of genetics. Unfortunately, this isn't where the story ends. We're going to learn all about these non-Mendelian inheritance patterns in our next lecture. I hope you enjoyed the video. If you have questions, let me know.